So I guess we can get started. It is 10 a.m. So good morning, everybody. My name is Dustin Ellis. I'm one of the foresters with Hennepin County, and I'm also one of the board members for MnSTAC. And this is the holiday forum. So normally we'd have a potluck after this, but seeing as we are virtual, you'll have to provide your own pie this year. And today, well, first I want to give some thanks to the University of Minnesota for all the hard work they've done this year with the virtual forums and putting these together. I know some of them have been very time intensive. So a big thanks to the University of Minnesota for this, so that we are still able to have our forums during a challenging year. And into the future, we're gonna most likely have to keep doing these virtual forums for a while. So hang tight, we will be live again sometime soon, hopefully. But today, for our holiday forum, we've got a real treat for you. And today we've got Dr. Keith Gilland from the University of Wisconsin Stout. He's been working on chestnut research since 2003, examining chestnut ecology and naturalized populations, its regeneration needs in mined land restoration, and its role in disturbed ecosystems as a wildlife tree. Fun fact is he recently completed his wildland firefighters certification. So this session is going to be on how the American chestnut played a significant role in Appalachian ecosystems prior to its near extirpation by an introduced fungal pathogen. Current restoration efforts focus on breeding, transgenics, and multiple pathogens in a changing landscape. Those efforts, challenges, and successes will be discussed along with future opportunities for research and involvement. So with that, I am going to turn it over to Keith.
Yeah, well, thank you very much, and thank you all for coming to this online event. So, yeah, what I'd like to talk about today is some of my own work and some of the other work going on with chestnut out there, and kind of give a run through of some chestnut history and ecology, and yeah, we can entertain any questions that you might have, and hopefully I'll maybe have some answers to you. So. Yeah, with it being December, it's chestnut season and time to roast them on the old open fire and all that stuff. So no better time than the present. So a little bit about me and my sort of background with chestnuts. I started working on them around 2003 or so when I was an undergrad at Miami University in Southwest Ohio, and I started working with a faculty member there, Carolyn Kiefer, who had been working on chestnuts for a while and still does. And she had a grad student at the time, Janice Bauman. She was a doctoral student. She had come from Bill McDonald's lab, and he had worked on chestnut hypovirus. We'll talk a little about the hypovirus later. So she had worked on some blight control techniques previously, and she came to Miami to work on chestnuts and mine land reclamation. So these are a couple mine sites in southeastern Ohio that I worked on as an undergrad, and these were older sites that had been mined in the 50s and 60s and kind of abandoned, and we were trying to get chestnuts growing on those. And then my undergrad thesis project was done down in West Salem, Wisconsin, at a stand that had it was this disjunct stand that had been introduced from a handful of nuts planted by. The story went from a family member that was from Pennsylvania and came up here and said, you need chestnuts, I'll plant a few. And these trees had naturalized throughout this 100-acre or so oak hickory stand. And this was 2005 summer. This was the largest surviving American chestnut tree. It is no longer surviving. It was in pretty big decline when we were there even then, but it was really a massive tree. It's kind of cut off in this picture, but this is about a 6 or 7 foot basal diameter tree here. So this branch goes down. And I'm a relatively normal sized human being for scale. So I messed around with chestnuts and minelands and this nut production stuff as an undergrad, and then I went to Ohio University, which is different from Ohio State University. That's in it's in Southeast Ohio, and I worked on uh, chestnuts and mineland reclamation there. And at that time, we were working on methods of reclaiming newly mined lands and sort of the ecology, and but then also chestnut introduction. So uh, I totaled it up one time, and I think we planted somewhere in the range of 5 or 6 thousand trees for different projects and experiments over the time there, from bare root trees and planting nuts and different hybrid lines and different experimental treatments. And so worked on that for a while. We also had a couple projects on some older mine sites where we were doing some soil ripping and compaction relief and trying to plant chestnuts there. So did that and got my PhD in 2012, and then went to another school in Ohio, and I taught statistics there for a few years as a visiting assistant professor, and that kind of left some time to follow up on things that I had gotten started with on my dissertation that I kind of wanted to keep going. So kind of kept up with the mine land stuff and started a few projects in Southwest Ohio with chestnut restoration and doing some soil treatments, namely some soil acidification. So trying some elemental sulfur and a couple other treatments designed to acidify the soil there. We kind of had the idea that if we had the right texture and drainage and we fixed the acidity issue, we might be able to grow chestnuts. So kind of worked on that for a while and got involved in a couple projects dealing with trying to plant chestnuts in old pine plantation clear cuts. So Southeast Ohio had a lot of civilian conservation corps pine plantations that, well, I think everywhere has a lot of old civilian conservation corps pine plantations, and mainly white in Virginia, and a lot of the areas those were planted in, those trees aren't quite native to theirs. They don't do much regeneration, so state forestry was looking for something to do with those sites as they were getting kind of to the end of the the pine lifespans there, and like, well, it would have been sort of a mixed mesic deciduous forest, and well, chestnut might be a good part of that. So I kind of feel like I have a habit in my life of finding research to do in places that just kind of absolutely suck to go to, whether it's recently finished mine sites or pine clear cuts or like I feel like I need to get myself involved in like some mature second growth forest ecology. That's what I need in my life. So we work on we've been working on the pine plantations and kind of following up with a lot of this chestnut and mineland stuff to see how it works as a part of a developing new forest. So I've been doing chestnut stuff for 17, 18 years, which may or may not be a long time. It feels like a long time, but yeah, still kind of into it and wanting to do a little more up in this area now that I've found myself sort of transplanted up to the upper Midwest area again. So a little bit of an overview of how I kind of want to go through things. We'll talk a little about the historical chestnut ecology and its importance and its decline after the blight. Then we'll look at a few of the early control efforts from what would have been just straight up arboriculture techniques at the time. And then we'll get into this. The American Chestnut Foundation has called this the three burr strategy. And there's the biocontrol, the back crossbreeding, and the biotechnology realm, sort of three avenues to go about tackling the problem of the chestnut blight. So we have a fungus that kills the trees, and well, we got to do something about it. So we'll talk about sort of what those look like and where they are currently, and then we'll sort of circle back around to some of the research possibilities in ecology, and if there's time, sort of do a few more kind of fun pictures of stuff I've been involved with and still kind of work on related to chestnut stuff. So, um, you know, introduction for depending on how sorry familiar you might be with the chestnut. It was widespread throughout the Appalachians. There's some dispute over how dominant it was. You see some figures bandied about like 25% of the basal area, and like, well, that might be based on some 25% of the basal area in stands that were really good spots for chestnut to be growing. Also, at certain times, it's a pretty prolific stump sprouter, even for stuff in the Fagaceae tends to be pretty stump sprouty, and my experience with chestnut has been it's especially stump sprouty even for the family. So some of those figures from around the late 1800s that say there was tons and tons of chestnut are potentially also kind of maybe an artifact of a lot of the land clearing that went on in sort of the middle 19th century. So not to say that it wasn't an important and widespread species. Um, some of the wilder figures you see about its abundance might be a little overstated, but and those are to me that kind of makes sense that at a time in our history we made a lot of area that was really good for chestnut to grow in, and it really exploited that niche, which then really made the blight bad because we had made conditions to have a lot of chestnut. And then when we killed it, we killed a lot of what we had. So it has the reputation of being a very, very prolific and reliable nut producer. And this was my undergrad project was, well, by the time people started really doing the work to put a lot of numbers to this, the species was mostly kind of extinct. So we had a lot of figures for like how much an orchard grown chestnut would make, nut production wise. But we were fortunate to have this naturalized stand up in West Salem at the time that looked pretty much like what a sort of stereotypical oak hickory chestnut forest may have looked like in the Appalachians at the time. And so we went up there and did our study, and this was based on sort of nut counts and then sort of extrapolating up to pounds. And so it does outproduce its sort of closest competitor, the red oaks, and it's also pretty reliable. They're not a masting tree. They do their nut production pretty reliably each year. They're a late spring flowering species, so they flower pretty late in the year. So their reputation is that they're pretty immune to some of the late spring frosts that tend to not knock back the hard mast crop in other species. So important wildlife species. There's a lot of sort of anecdotal evidence out there about species that were lost in conjunction with the chestnut declining. Again, it's kind of hard to tell, is that, directly related to chestnut? Is it, influenced by chestnut? Is it related to all of the other land use changes that were going on at the time that chestnut was declining? But um, it definitely does make a lot of nuts, and they're nutritious. They're palatable. They're pretty low in tannin content. So they're kind of a food of choice. And then on the timber side of things, is sort of um, sort of a bulk structural wood. So telegraph poles, railroad ties. It has a reputation as being fairly rot resistant. So if you go down, Blue Ridge Parkway area, there's still a lot of wood chestnut stuff down there, whether it's old split rail fences, ties and poles. You do see it show up as a furniture wood in some places. It's pretty light. It's pretty easy to work. I have a few chestnut turned wood pieces that were made out of old reclaimed stuff. It's a pretty wood. But yeah, it had so it had this, nuts as a cash crop. It was a timber crop. They talk about it being a, cradle to grave species in some places. So important, wildlife species, economic species. Um, it has a decently high, tannic acid content in its bark and leaves. So, the extraction of natural tannins was another part of its industry. So, um, its reputation as being an important tree is deserved. So that's a little bit of the history. Then the blight. So um, blight first started to show up, or was first observed in the early 1900s, and it was first noticed at the Bronx Zoo, and it spread outward from there, and it infected the entire range by 1950 or so. So by 1950-ish, we were down to really only having root sprouts remaining, species more or less functionally extinct by then. So it is a fungal pathogen, Cryphonectria parasitica. It causes these perennial necrotic lesions on the bark of young trees. You get these sunken cankers on the small, smooth barked twigs. On larger trees, oh, sorry. On larger trees, you get these larger cankers, and you see the sprouting below these. So where you find mature trees now, what you generally see is these large orange cankers above, and prolific sort of stump sprouting, or if it's higher up on the tree, this sort of epicormic branching below that. The mechanism of death is these these uh, these mycelial fans beneath the bark that are related to xylem clogging, inducing the production of tyloses, and also just the degradation of the cambium under the bark. So that's kind of the the blight species. Originally Endothea parasitica, now Cryphonectria parasitica, and widespread throughout the range. It has a few alternative hosts, and mainly in the Quercus species. Scarlet oak is the one I'm most familiar with as being the bad guy, but it's also been observed on post oak, Quercus stellata, and also in the live oaks. And as far as white oaks, some people say you find it on the white oaks. I haven't found any written proven evidence of that, other than like, well, people that I would trust say it happens, so I wouldn't discount it out of hand. But the scarlet oak alternative host is the one that has generally been the biggest concern, or most well known alternative host for it. So as far as some of the what we saw after the decline. So, like I said, we had the entire range affected by the 50s, and depending on where you were, mostly you saw a change over to a mostly oak and hickory dominated ecosystem. In some places, in some of the more mesic coves throughout the Appalachians, you saw a switch over to hemlock, but mostly, what we saw was a switch from a sort of oak hickory chestnut matrix to an oak and hickory situation. And now, sort of as an aside, there's that concern of like, well, those are sort of switching to more of a maple dominated forest in a lot of those regions. So, like whether or not we would see, it's sort of academically interesting, whether or not we would have seen, chestnuts being lost if they were still part of these forests. So we would be seeing them lost as part of the oak regeneration failure, or would we see them as a sort of more competent player with the sort of mesification and switch over to maple in a lot of those same forests? It's not something we can really answer, but it's also one of the things that I think is very interesting to think and talk about. So we saw the decline, and there's been a lot of interesting papers put out about this over the years, sort of what happened, and trying to piece together some of this history through archival evidence. But well, the species again largely lost by the 50s. So looking at like as the species or as the blight was moving through and happening, um what were people doing? And at first glance, well, it's a fungal pathogen. So like, well, how do you treat fungal pathogens? And a lot of the initial treatments were what you would do, prune and burn diseased branches. So if you have a fungal infection on a branch, you cut the branch out, you burn it, and well, that's what you do. And it had worked before, why wouldn't it work now? um Spraying, sort of bigger prize specimens with Bordeaux mixture, copper sulfate. So again, it's a fungal pathogen. So um, maybe the things that control fungal pathogens in other species would work on it. It didn't work. It's since it's mainly living and going on under the bark, so sort of surface treatments didn't have much effect on it. But some things that were tried. There were also some um, large scale removals to sort of stop that disease front moving as it was moving, southward through the range. And this was sort of a combination of like, well, maybe it'll stop the blight, but also, some timber speculation with, we're kind of at the depth of the depression, and, hey, why don't you sell this timber before it's not worth anything? So, there's always the also classical like, oh God, what if we cut down the one that was resistant to the blight? And like, well, yeah, maybe that happened, but it's not like it's something you can do anything about now. So yeah, this is a little bit of an overview of there were some attempts at the time to stop the disease using pretty classic plant culture techniques, and they weren't effective. But yeah, so we lost the species, and so that kind of brings us to well, Castanea mollissima, the Chinese chestnut, exists, and there was a movement in the 50s and 60s to look at, well, we're losing Castanea dentata, the American chestnut. Why don't we plant Castanea mollissima, the Chinese chestnut? And the issue with Castanea mollissima is it doesn't really grow like an American chestnut. So this is a chestnut orchard, grown for nut production. This is in Eastern Ohio. This is the Empire Chestnut Company, and they sell food chestnuts online. They sell bare root and potted chestnuts, over the internet and through the mail. And sort of taking a look at these trees grown in this orchard, they have a sort of orchard tree habit. And so, there were attempts initially to, plant these Chinese chestnuts out into the forest, and yeah, they don't really reach the canopy, so they don't end up competing with, oaks, hickories, walnuts, North American, forest canopy trees. So that effort didn't really work out, which isn't to say that there aren't Chinese chestnuts that could live in our forests here. It's the sort of matter of if you want to learn about Chinese chestnuts in their native habitat, there is that literature out there, just most of it is largely in, Mandarin. And there was an envoy sent to China from the American Chestnut Foundation a few years ago, and it's sort of a matter of like, well, do Chinese chestnuts just not grow to the canopy, or do all of the Chinese chestnuts we grow in America look like this because these are the Chinese trees we have selected to grow because we wanted to grow them in orchards? And it's probably somewhere in the middle. So Greg Miller, who runs Empire Chestnut, has had some success breeding some pretty decent timber quality, Chinese chestnuts, or just half American, 50% hybrids. And so the potential to grow, Castanea mollissima as a forestry is out there. It was kind of tried, and it didn't immediately work out. So it was kind of abandoned early on. And there were some other just sort of fun and fantastic attempts at dealing with chestnut blight that I've kind of run across. I was in West Virginia buying some trees early on in my graduate work. They had trees that had grown from, they were trying to irradiate seeds. And like, well, maybe like it's the 50s and it's radiation. Maybe that'll fix it. And so there were some offspring that, well, we had these irradiated seeds we grew, and well, that didn't really work either. So, some sort of throwing a lot of things at the problem and trying them out, and then nothing happened for a while. So this is kind of the point, like, well, and then it was kind of the early to mid 60s, and initial cultural stuff hadn't worked, and some early, hybridization attempts hadn't worked so well, and then things kind of, settled onto the back burner for a while. And then we kind of move forward, and we get into the 80s, and there's kind of a resurgence of interest in chestnut stuff. And so this kind of moves us into the current ongoing efforts, which is this three burr thing: the biocontrol, the back crossbreeding, and the biotechnology. So the biocontrol efforts, these had their origins in looking at what had happened in Europe. And there's a European chestnut, Castanea sativa, and it had been more or less wiped out by the chestnut blight, but there was an observed recovery of the chestnuts in Europe, and that was due to the development of these hypovirulent strains of the chestnut blight. And so these hypovirulent strains, these are strains of the chestnut blight that are infected with the virus from the Hypoviridae family. And so basically what these do is they infect the blight fungus and make it less lethal. And so what we see is the cankers still form on the trees mostly, but they're not lethal to the trees. So you'll get a canker, it'll heal over, the tree will be kind of gnarly, but it'll keep growing and living. And the virus is transmissible through the cytoplasmic connections that fungi make, and it's fairly easy to do in the lab. And the issue has been in the United States, the large scale introduction of these hypovirulent strains has been kind of difficult. So as I was saying, at the time the meeting started, Bill McDonald did a lot of this in Michigan, and then also down at the West Salem, Wisconsin stand. And sort of in general, what they would find is you could introduce the hypovirulent strains at sort of the canker level, but we weren't seeing it. I was there at the time, I saw it. This wasn't a thing I worked on, but what they would see is that it wouldn't generally move throughout the stand and transfer from tree to tree and canker to canker in the way that it did in Europe. So this kind of trailed off. It had a lot of promise because it had worked in Europe, but we kind of saw it here and like, well, our fungal, our transfers of this virus don't work so well. And it turns out that it seems that in North America, there is a lot more genetic diversity in the realm of these vegetative compatibility genes in North American strains of the chestnut blight that didn't exist in Europe. So the European chestnut blight is fairly homogeneous, whereas in North America it seems to be quite a bit more diverse. So the genes that control whether or not you can make your cytoplasmic connections, much more diverse in North American chestnuts, and that kind of hampered these efforts to get the blight hypervirulent strains of the blight to move around. Um, so is it dead? Nah, not really. So this is one of these things where the molecular revolution potentially has promise to reinvigorate this hypovirus and biocontrol as an option in the United States. So um, there are lots of other hypovirulent or a lot of other hypoviruses out there that might be candidates for use. So the CHV1 virus is the most commonly studied one, but there are at least a dozen or so others that exist that aren't as well studied. So, sort of option one is like, well, maybe there are other applicable strains of hypovirus that could work. So, there is also the possibility to integrate hypoviral DNA into the chestnut blight fungal genome. So that may also be something that would help spread this hypovirus throughout North American um, strains of the chestnut blight. So, there might also be the possibility to uh, disrupt these vegetative incompatibility genes in strains of North American chestnut blight in such a way that allows them to make those cytoplasmic connections that now are kind of stopping the spread of the chestnut blight. Um, so sort of in summary, like biocontrol had a lot of promise and it worked okay, and there are still people that have large surviving American chestnuts on their property that mud pack their cankers with these hypervirus strains and sort of treat large individual chestnuts this way, and it keeps them alive and it keeps their genetic material in the mix. So to say that biocontrol didn't work is not entirely true. It works in some cases, and there's potential to use it as one of the weapons in the arsenal in the fight against the chestnut blight. So there are some options out there, and there are some people working on these, and there are some citations for the people working on these at the end of the talk here. So that's a little bit of the biocontrol part. Is maybe we can make the chestnut blight less lethal, and maybe that'll work. So the the angle that got the most attention for the longest time has been the back crossbreeding program, and that's what the American Chestnut Foundation has been working on for the last 30 years, 30 years or so. And so the back crossbreeding program is a very traditional plant breeding program where we have an American Chinese cross, so dentata and mellissima, screen the offspring for blight resistance and American characteristics, back cross that generation with an American, continue this back crossbreeding program with Americans, and screen at each step for blight resistance. And the blight resistance screening is sort of just a canker assay with use a cork board to punch a hole in the bark and give it a smear of blight inoculum, and then score them based on severity of the canker that develops. And the smaller the canker that develops, the better. And then the sort of physical characteristics are mostly bud and twig characteristics. So American buds are typically, sort of longer and slender. So are the twigs. The Castanea mollissima twigs are usually a little stouter, and they have some ribbing to them that look a little different. And there's also quite a bit of uh, kind of gestalt in the, like, does this young tree look like an American or a Chinese? And um, it was the sort of thing, after doing it enough, I felt like I had a, not to say that I had the expertise to be a critic of the, blight program, but I remember going to buy trees and them being sold as these are our pure Americans, and kind of giving them the side eye of like, no, they're not. That's a Melissima twig. And so the initial early blight or breeding screening is done on those bases. And so um, after the third back cross, then there is proposed 3 generations of intercrossing to hopefully arrive at a third back cross, third intercross tree that is um, both blight resistant and highly American in characteristics, and hopefully true breeding as an American looking tree. So that's been the Chestnut Foundation back cross breeding program. And these trees are now out in test plantings where there's sort of fields of the various parental strains and American parent, progeny tests. So that's where most of these are now, are out in the field progeny testing. And then at the same time, there's the um, state chapter programs that are going on. And so there are 16 of these state-run volunteer-run state chapters. And um, what these chapters do kind of varies from chapter to chapter, and there are some that have had sort of concurrent breeding programs going on for more or less as long as the main Chestnut Foundation program has been going on. Um, some of them have been focused on just sort of finding and propagating and keeping chestnuts alive in the state to keep the genetic material alive. And some in Ohio, I would have said we were much more involved in planting trees. So it kind of varies from state to state. Like I said, some do a lot of breeding, some do a lot of planting, some do a lot of conserving remnant populations within their states. So these have been a major source of strength for an organization like this, just having the people power and the volunteer power to keep these progeny tests that are going on. Just simple things like keeping them brush hogged and weeds sprayed and deer fencing up, and all of the things it takes to run a big sort of research and restoration project like this. So kind of where we are with the breeding program now. So um it's the breeding program is progressing. Unfortunately, it kind of turns out the blight resistant in these BC, the third back cross F3 generation hybrids, has been um, more variable than had been hoped for. So that may be underlain by there are probably quite a few more genes involved in blight resistance than were initially planned for. More genes on more chromosomes than was planned for and would be ideal. So that's going on. And there's also this tension in practical terms of um, moving forward with rolling out trees or doing more generations of intercrossing, um, while also waiting to see what the full story is. So, the problem of trying to make a sort of 100-year prognostication based on a 7 or 8 year old tree. So that's kind of where the breeding program is now. It was going to be the thing that saved everything, and it's maybe needs a little more work than had initially been planned for. So things that are ongoing with the breeding program uh, are trying to introduce more sources of resistance. And so when we say more sources of resistance, we mean um, either Chinese or Japanese parents. So most of the breeding program now, the Chinese parents, or the Chinese source of resistance is coming from just a few parents. So there's kind of a movement to recognize that like maybe we need a more robust source of resistance. So um, that's uh that's an ongoing part of the breeding program. And then sort of the summary, there was an external review of the science of the breeding program conducted in 2018, and this has been part of a sort of decadal review of the breeding program. And so in the previous slides, expanding the resistant sources beyond the original, they call them the Clapper and the Graves trees, and they're named that based on the places they came from. So this is a Chinese tree that was on Clapper's place. This is a Chinese tree on Graves's place. So um, expanding resistance sources, so getting more Chinese parents into the mix. We've done a good job of keeping American parents in the mix with the idea of genetic diversity, but there's also the issue of like maybe we need some more diversity in the other side of the hybrids parents. So looking at genomic methods to screen for blight resistance. So finding out if there are any genomic markers we can use earlier on to say, yeah, this individual is gonna be blight resistant or not, versus sort of growing them a few years, doing the stem assays, growing them a few more years to try to turn the breeding program around so the generations move a little faster. And then as sort of what I as a plant ecologist am more interested in is getting more of the ecology and silviculture of chestnut sussed out. It's a sort of tree that um, isn't grown as much as other trees are. So there's still some gaps in the knowledge of, if you want to grow big, good looking, healthy chestnuts, how do you do that? Not completely known. There's a lot of conflicting ideas and hypotheses out there about what they like in terms of soil chemistry, what they'll tolerate in terms of soil moisture. There are people that say that they will grow in a pile of gravel with no water ever, versus like, well, some of the biggest ones ever found were growing in these really mesic coves in the Appalachians. So is this idea that they're a xeric species something that's left over from, well, the ones we find mostly still growing out there are just ridgetop trees that haven't been out competed. So of course we find them there. So there's a lot that isn't known about what they like to grow in, and how to grow them, and how to best propagate large quantities. So if you want to grow lots of chestnuts for sort of the people in your nursery, what are the best methods to do that? A lot of stuff isn't known. And so, what's immediately coming up is to do some work at the Meadowview Research Farm, where most of this has been done, and do some culling of a lot of the older trees, and try to focus in on some of the most blight resistant. And this restoration project is a human thing. It can be tough to kill your trees if you think they might, like, this one might still be good. So I plan to sort of do a more ruthless sort of, we're only going to keep the best of the best in the breeding program, and the potential also rans, like, those need to go. So that's coming up. And then um getting more of these you know third backcross, third intercross trees out into the test planting. So, when I first started doing chestnut stuff, the final trees coming up were always like 5 or 6 years away, and it's like 17 years later, and still the good trees are 5 or 6 years away. And that's not a like, as much as that sounds like a dig at the breeding program, that's just the realities of trying to breed long-lived trees. They're not annuals. It takes a while to grow them up and for them to reach reproductive maturity and run a program like this. We want things to happen on human times that need to happen on ecological times against a backdrop of a disease we spread up that would have only spread on like geologic time. So keeping in mind that we have this time frame that we're trying to speed up, and maybe we haven't sped it up as much as we would have liked, but we've still sped it up quite a bit. And that kind of brings us to the biotech part, and this is what's gotten the most attention lately. And this was sort of something being talked about in possibilities and hushed tones and um, sorts of really big ideas when I first started coming around, and it has become reality. And so the overall deal with the transgenic chestnut trees is they've been transformed with an oxalate oxidase gene from wheat. And so the sort of method of blight resistance here is the sort of physical damage done to the trees is from oxalate production from the blight fungus, leading to this breakdown of the cambium and xylem. And so if you can neutralize that, well, you can sort of neutralize the damage being done by the fungus. So the fungus still infects the trees and it still does what it does, but the tree has a method to neutralize the oxalate that the blight fungus is making. So not natively produced in any Castanea species, this oxalate oxidase gene, the OXO gene. And so the initial rounds of these transformations were done in 2006, and this is all at the SUNY, the State University of New York, at their forestry program. So this is coming out of there, and they did their first round, and it wasn't actually super successful. So they got the genes in the trees, but they didn't really have the desired results. And between 2006 and 2012, in the next 5 years, they focused mostly on improving the promoters for those genes to actually get the trees to express them. And so once that was squared away, the trees produced now seem to be highly blight resistant. And so there's been some other really good work that's gone along with this that has carryover to the breeding program, which is the small stem assays. So the ability to screen for blight resistance on these 1-year-old stems that was developed to do this on the transgenic trees, but it has the ability also to really help out with the more traditional breeding program that you can screen for blight resistance in sort of the first year. So also some work with tissue culture. So propagating trees via tissue culture is also something that's come out of developing these blight resistant trees. So that's kind of we'll talk a little bit more about where they are regulation wise and some of the environmental testing going on. But the overall deal with this transgenic tree is it has a serial gene to produce oxalate oxidase, and that's the sort of thing that neutralizes the chestnut blight in such a way that it's no longer lethal to the tree. So kind of where these are, tree has been developed. Now it's into the sort of regulatory approval. And this is all of the well, if we want to introduce this transgenic tree onto the landscape, how do we try to ensure that this is, safe in terms of its interactions in the ecosystem? So, um, there are trials with pollen preferences and effects on aquatic invertebrate species, so mayfly larvae, not having any negative effects. So, pollinator feeding studies haven't seen negative effects on growth and survival. There are tests ongoing and have been ongoing with potential allelopathy, so germination effects on co-occurring species. So, kind of traditional grind up the plant matter in the litter and throw it into your pot and see what happens. There's some papers out there that talk about chestnut being an allelopath, but I don't know if you kind of find like I feel like a lot of things get tossed out there as a potential allelopath, kind of based on the like, well, these are a fairly dominant species. Maybe they're an allelopath. Well, probably not. But there's been those studies. So sort of looking at effects on invertebrates, on germination studies. So these kind of go into the case. Vertebrates, so particular vernal pool species, so tadpoles and salamanders. So, again, studies of doing these leaf litter and water studies to see if there are any effects on that front. What they've mostly been finding is any sort of uh, interactions in terms of leaf litter decomposition. The transgenic trees seem to behave mostly like the um, hybrids and pure American trees, rather than there being sort of differences between pure Americans and the hybrids and the transgenic trees. And so some of the work I did as a grad student looked at these hybrid lines, and what we generally saw was ecologically the Chinese trees were kind of over here in terms of a lot of leaf characteristics and decomposition and breakdown, and then the hybrid stuff and the pure Americans were kind of over here in your when you make your bar plots and your sort of pairwise tests and your ANOVAs, the hybrids and Americans would generally be kind of over here. And then what we're finding is the transgenic trees are clumping over with the pure Americans and the hybrids, and that's a good thing to see that ecologically they behave similarly to the pure Americans and the hybrids, rather than kind of being off somewhere else or off with the Chinese trees. Mycorrhizal fungi associations are another thing. Not really seeing any difference there. So the colonization rates and sort of what can be screened for in terms of species of mycorrhizal fungi seem to be the same. And sort of anecdotally, my experience with chestnuts and mycorrhizae was we generally found it was more of a site-specific thing than anything else. Whether it was treatments we were doing, so we would do a lot of inoculate with different a few different species of mycorrhizal fungi, and what we would generally see was the environmental filters were stronger than the treatment, or the site filters were generally stronger than anything else we did. And so it seems like the mycorrhizal fungi associations are um, pretty similar across the hybrids and the pure Americans and the transgenic trees. So sort of where it is now as far as regulatory. So the the public comment period for the the release of the tree closed a few weeks ago, and I haven't seen the results of that yet. So they were up for regulatory approval through USDA, APHIS, and if that gets the okay, then the uh, transgenic trees will go out for these sort of fuller scale test plantings, so get planted out into fields with themselves. And so also on the genetic front, the further mapping and identification of blight resistance candidate genes in the Melissima and the hybrids. So the chestnut genome has been sequenced, but it hasn't been fully mapped and identified. So we kind of know we know the orders of the A's, the C's, the G's, and the T's, but the what it all does and where they all are isn't fully known. So there are some candidates that are done from sort of screenings to find some potential candidate cis genes. So like looking at some genes expressed at the actual cankers in Melissima and dentata, American and Chinese. So sort of looking at the genes expressed in the two might give you some possible candidates. Like, well, we don't really know what this gene does, but it's active around these cankers in Chinese chestnuts and it's not in American, so maybe that means something. So there's not just the transgenic part that's important, but there's also all of this other associated work that allows us to see what's going on and potentially identify what is it in the Chinese chestnut genome that makes them blight resistant. So that, something hopefully coming up soon. And so as we get sort of out of time, sort of where I am, I'll sort of close with this. This is kind of what I'm into still, and it's Mindlands and it's old pine clear cuts, and it's places that need trees growing on them. And if chestnuts might do okay on them, I say plant them on them and see what happens. So these are some old mine sites in Ohio, couple years apart. This is another old mine site. This was a grassland that had been sort of ripped and plowed and disked, and these are the pine clear cuts that, again, just a miserable place to be in the summer. So it turns out when you clear cut all your pines, they fill back in with rubus. Who could have imagined? And so with that, I'll stop sharing, and we have time for any questions or anything folks might want to talk about. I would be always happy to sort of talk chestnuts. Let's see if I can figure out how to stop sharing. There we go. Oh, there we go. No. Okay. And Ryan, do you have oh, there you go. Okay. All right. So I see a few questions here. So I'll start. Could it be on the native historic range? Yes, I hope so, and I think so. So I think the trees over in La Crosse sort of show that it's definitely possible, and they would do quite well. It's something that I would like to try to do more around here. I said there's the firefighter training thing. The thing I've gotten myself mostly involved in up here is a lot of prairie and savanna restoration, but I'm doing a lot of work on some old sand and gravel mines, and they kind of have seem like ideal candidates for some chestnut restoration. They're sort of sandy. There's not much going on in a lot of them in terms of native vegetation. So those seem, as a landscaping guy and a sort of general forest ecologist, like these look like they could use some chestnut. So yeah, I think there's a lot of potential for that to happen. Castanea crenata. To remind me, so there have been, I know with sativa, and it kind of, with their sort of lack of blight resistance, didn't really do much. Crenata, is that is that chinkapin? Or is that Japanese? Okay, Japanese. So that has been to my knowledge, no, but it's one of those things that's in the like, we should probably think about that as a way to, look for other sources of resistance. And, there's and again, I feel like sometimes I'm kind of overly negative about the, the chestnut program. And, it's easy in hindsight to be like, oh, this has so much tunnel vision. They only focused on a couple of these Castanea mellissimas, but it was 35, 40 years ago. And it was like, well, this, seemed like a reasonable enough thing to try. And, then as it sort of gets its momentum going, you end up that, well, here we are 35 years later, like, oh man, I wish we could go back 35 years and do something different. Yeah, so the Ozark chinkapin, there are, to my knowledge, not much in the way of blight resistance breeding programs going on. There are people interested in conserving the species and um, trying to sort of keep at least semi-resistant varieties under cultivation. So, finding the ones that are still surviving and sort of keeping them growing and collecting seeds off of them, collecting cuttings off of them, and keeping those in cultivation in some way. I
Hey,
guess I didn't quite get to it in the end here. There's another problem in kind of circling around to like maybe planting outside of the original range, that up here we would maybe be free of the other problem with chestnuts, which they're attacked by a Phytophthora root pathogen in the southern parts of their state. So as far as spacing, in most of the orchard plantings and test plantings, we go for, 15, 20 feet for them, to give them that'll kind of get them to the size they want to be in. And, their crowns will touch at that point, but that's generally what most of the progeny test out plantings are done at. But up here, the idea of being freed from sort of the Phytophthora root pathogens is appealing, and so those kind of started showing up down in Georgia mostly. And when I was working initially on the mine stuff, we were always sort of working under the assumption that like, well, these oomycetes like it wet, and these mine soils, they drain so well that won't be a problem. And it's also too cold up here, but 15 or 20 years of a changing climate kind of makes, Kentucky a lot warmer and wetter than it used to be. So even in some of the places that we thought we were kind of free from the Phytophthora problems, they're showing up. So the options there are like, well, we could breed a Phytophthora resistant tree concurrently, or develop a transgenic Phytophthora resistant tree, or well, maybe we could grow them in Eau Claire, and maybe there won't be a Phytophthora pathogen in Eau Claire.